Hello, my name is Winston Koh, the Chairman and CEO of Genervon. I'm a businessman and an investor who embraces entrepreneurial challenges. I've chosen to share our latest bioinformatic and clinical findings, including confidential list of ALS, Parkinson's, and Alzheimer disease-associated genes modulated by our multi-target drug GM6 during the J.P. Morgan Healthcare Conference in San Francisco. In his book, 0 to 1, Peter Thiel said, "We have given up our senses of wonder as secrets left to be discovered. We can't find secrets without looking for them." He also said, "We are within reach of ambition so great that even the boldest minds of the scientific revolution hesitated to announce them directly." Today, I am also hesitating to share with you the secrets Genervon has discovered. 20 years ago, I was looking for worthy investment opportunities. I realized that something must be wrong with the uniform failure of clinical trials looking to find cures for various nervous system and neurological diseases and disorders. It became obvious that CNS disorders are very complex and that any single target drug focusing on some pathogenesis cannot cure the complex multifactorial nervous system diseases. Instead of custom designing man-made molecules to target specific defective genes, it seems more logical to find the endogenous regulator, which are responsible for embryonic stage development and can regulate and repair a diverse set of disease pathways within the highly complex human nervous system. I asked a team of scientists to find a novel endogenous embryonic stage regulator that controls the development of the nervous system, including the differentiation of embryonic stem cells, the monitoring of the cellular stress signals from malfunctioning genes or misfolded proteins, and the discharge of specific signals through various pathways for the repair of the malfunctioning genes and proteins. Our team discovered a critical regulator of human nervous system, which we named GM6. We then spent over 10 years developing GM6 through many preclinical studies, including in vitro and animal models. Here is the 104-page executive summary of the investigator brochure. Each experiment report summary took less than a quarter page in the investigator brochure. GM6 has demonstrated its safety in preclinical studies, as well as in 1 phase 1 and 3 phase 2 clinical trials in ALS, Parkinson, and ischemic stroke. GM6 is a small peptide delivered by injection. It can pass through the blood-brain barrier and is very safe with few to no side effect. Today, I'll share the bioinformatic findings suggesting possible GM6 mechanism of actions for ALS, Parkinson's, and Alzheimer's disease. So, what's the secret? Genervon's research discovered and confirmed that GM6 modulates thousands of genes in the nervous system. This suggests that GM6 is a master regulator that affects this extremely intricate network of disease-related pathways in the central nervous system. Here's a short summary of our discovery. The mechanism of action for GM6 appear to involve many different pathways, and that collectively, the combined action serves to promote homeostasis and correction of multiple disease states. GM6 does not act solely as an agonist or antagonist. GM6 is not a cocktail of different molecules. It is an endogenous embryonic stage tyrosine kinase motor neuronal trophic factor regulator. It binds to insulin receptor, IGF-1 and IGF-2 receptors of the human nervous system. It can up and down regulate the same genes depending on whether the disease gene is below or above the normal range, respectively. It also regulates through multiple signaling pathways in response to distress signals. DNA microarray were used to compare gene expression in GM6 and control treated SHSY5Y cells. GM6 regulates 1,259 to 2,207 genes, within which GM6 regulate 89 to 238 ALS-associated genes, 48 to 68 Alzheimer disease genes, and 46 to 59 Parkinson disease-associated genes. GM6 also modulates genes associated with other neurodegenerative and neurological diseases. The chart shows the reason why ALS is a more difficult disease, requiring modulation of 238 diseased genes, 4 times more than Alzheimer's disease. ALS is a very heterogeneous and multifactorial fatal disease. The combined effects of GM6 on these 89 to 238 genes represented a coordinated systemic response that can modulate ALS disease progression. GM6 restore cellular homeostasis and modulate processes leading to motor neuron loss in ALS. These unique effects of GM6 would not have been possible through conventional drug design focused narrowly on a single target gene or pathway. Genervon's ALS poster is now published online, identifying all 89 ALS-associated genes modulated by GM6 more than twofolds. ALS genes whose expression was increased by GM6 were frequently associated with the cell division and mitosis, while GM6 repressed genes were often associated with mitochondria and the intrinsic apoptosis pathways. Our findings support the idea that GM6 can be a disease modification drug through multiple signaling pathways involving in ALS pathogenesis. Genervon is planning for an ALS phase 3 trial in 2017. Parkinson's disease is a progressive motor disorder caused by the death of dopaminergic neurons. GM6 strongly modulates the expression of 46 Parkinson's disease-associated genes by at least twofold. Genervon's Parkinson's disease poster is now published online, identifying all 46 Parkinson's disease-associated genes. GM6 repressed genes were largely associated with the mitochondrial protein complex and respiratory chain, and included genes associated with each mitochondrial complex. GM6 significantly downregulate expressions of most mitochondrial genes associated with Parkinson's disease. Alzheimer's disease is a progressive dementia characterized by memory loss, brain atrophy, and neuronal death. Genervon's Alzheimer's disease poster is now published online, identifying all 48 Alzheimer's disease-associated genes modulated by GM6. Our data suggests that GM6 may prevent Alzheimer's disease-associated neuron loss by attenuating mitochondrial dysfunction. For example, excessive reactive oxygen species generation, dysregulation of intrinsic apoptosis, limiting tau expression and accumulation, and bolstering APP catabolism. In conclusion, Genervon decided to share our confidential research findings, including the specific gene names, with the research communities. The goal is to try to persuade neurological researchers to go beyond the single-target drug development paradigms. This is a goal Genervon has been pursuing for the last 20 years with positive results. We encourage more scientists to explore this multi-targeted hypothesis. We welcome inquiry from researchers about our bioinformatic analysis or the need to access GM6 to conduct your experiments for treating disease of metabolic decline. We look forward to collaborate with researchers and pharmaceutical companies in these endeavors. Thank you.